If we have a look at the presentations in this session, we'll see that within the whole ecotoxicology space, we look at pollutants as they occur in the environment, how they distribute. Once they're in the environment, whether animals get exposed to them, if animals are exposed to them, whether they at subcellular level, whole organism level, and ultimately ecosystem level. In this session, we'll be looking at presentations at different levels of biological organization. And if I can just get the slide to move. Yeah, one would think we haven't been doing this for the past year and a half, but it doesn't work out any easier for some obscure reason. So what we'll be looking at is presentations related to the levels in the environment. We'll be looking at presentations that are related to uptake with possible bioaccumulation, and then effects that are at individual level and at other levels of biological organization. What I'd like to do is to give each of the presenters an opportunity to just give us a brief overview in not more than 2 to 3 minutes of what's the main take-home message of the findings of their study. And what we will then do is we'll carry on with questions directly to presenters, or just a general discussion as it goes along. If we look at the levels of biological organization and the whole ecotoxicological concept, I'm not going to follow the sequence in the program, but more what I might believe to be a more logical sequence is, first of all, to have a look at levels of pollutants in the environment. And the first presentation was by Marie Clementine and others looking at challenges related to antiretroviral drugs in African surface waters. Marie Clementine, are you on the meeting? Would you be able to give us a few, just some take-home messages from your study?
Yes, I'm in the meeting. Thank you. Hello?
Yes, please. Thank you. Go ahead, Marie.
Okay. Thank you, Prof. Vipna. My name is Marie Clementine Bamureke. I'm from the University of Johannesburg, the Department of Zoology. In our study, we expose fish to different pharmaceuticals. We expose to nevirapine. It is an antiretroviral, efavirenz, and the antibiotics sulfamethoxazole and trimethoprim. So in our study, we observed the pathologies even in the control group. Those pathologies should not be there. So when we took sample of water, we analyzed, also we found efavirenz in the control water. So that was a problem because even one of our article was rejected for that. So we decided to check then the water, the tap water and the borehole water we use in the aquarium, taking directly the sample from the tap, the different taps. So we took those samples from the tap water and the borehole water to the aquarium, and even at outside of the aquarium. So we checked for the different pharmaceutical. We test efavirenz, nevirapine, cifametaxazole, and trimetoprene. What we found is that efavirenz was the only pharmaceutical that was detected in the tap water only, not in the borehole water. And this consisted with the pathology we found in the control group, because those fish were exposed using tap water, not borehole water. So that was our main finding. So the highest concentration of efavirenz in tap water was 18.8 nanogram per liter. So we also tested the efavirenz, and other pharmaceutical in the distilled water from our lab, and we found that it wasn't detected in distilled water. So this is a problem because the water we use for the control doesn't seem to be like the control anymore, because it does contain pharmaceuticals. So our recommendation was that, like they do their dose routine checking of pharmaceutical metals and nutrient in the water, they should also add a list of some pharmaceutical which are persistent, seem to be persistent in the water, like efavirenz, so that there is a routine and a monitoring program for those pharmaceuticals. Thank you.
Thank you very much, Marie Clementine. So we won't go into questions yet. What we'll just do is give each presenter a 2 to 3 minute opportunity of presenting their take-home message. Right, so Marie Clementine indicated that antiretrovirals are present in the water that were used for control fish. So what happens when we have substances in the ambient environment and they get taken up by organisms? So it's sort of the lottery. Who's next on this? The next presentation that we'd like some feedback on is by Ashish Misra and colleagues, who looked at accumulation of and the subsequent uptake into fish from the Umgeni River system. Ashish, are you on the meeting?
Hi, how are you? I hope everyone is fine.
Thank you.
Okay, so for my master's, we studied the bioaccumulation of arsenic, mercury, and selenium in fish in the Umgeni River system. So we also looked at the concentrations in water and sediment. So we did this because the Umgeni River is one of the most prominent river systems in KZN, but it's also one of the most polluted, and many rural people use water from here as a source of water for drinking, and they use the river as a source of food because they eat the fish there, which they need for proper nutrition. So what we found was that water and sediment samples collected from the Nagal Dam, which is built along the river's run, had higher metals accumulation than the Inanda Dam. And when we looked at accumulation in muscle tissue, we found that those fish from Inanda Dam had higher levels of metals than that of Nagal Dam. So, and when I had checked the guidelines with the US EPA, it resulted in the Oreochromis mozambicus from the Inanda Dam being not safe for consumption for more than 1 serving per week, while Clarius caripinus and Copidon rendali were safe for consumption. That's about it from my end. I don't have anything else to say.
Thank you very much, Ashish. Keeping with mercury and moving a little further offshore, Hannes Erasmus and others looked at the levels of total mercury in different shark species along the South African coastline. Hannes, would you want to give us the take-home message from your study?
Morning. So yeah, we've looked at total mercury and elasmobranchs off the south and east coast of South Africa, and we found that elasmobranchs on the east coast has much higher concentrations of mercury than their counterparts in the south coast. And comparing this with a previous study conducted in 2005 to 2010, we also found that mercury concentrations increased over the past few years in the same species, and compared to global elasmobranch species of the same species, it's also several-fold higher. So I assume with Eskom and our power supply that's still predominantly coal combustion power plants, we are in the top 10 mercury emitters of globally. So the results that we found is correlating with that, and it's still increasing in elasmobranch species.
Excellent. Thank you, Hannes. Moving a little back into the land via the estuaries, Morelay Nell and colleagues had a look at metals in aquatic macrophytes in estuaries and their compartmentalization of these metals. Morelay?
Hi everyone, I'm Morelay. So we looked at 3 macrophytes in the estuary. 2 of them are salt marsh species, 1 is a seagrass species. The one plant is very popular in terms of research in Europe, and that's Spartina. So we know a lot about the uptake of metals in Spartina. So we know it accumulates a lot of metals in its roots, and that means it's a very good phytostabilizer. So it prevents metals from entering the rest of the food chain. The other 2 plants are only occur in southern Africa. So with those 2 plants, we looked at Salicornia tigitaria, which occurs in the upper intertidal, and that plant also accumulated a lot of metals in its roots, and it didn't have a lot of translocation to the above ground organs, which is a good thing in terms of phytostabilization. Now, if we compare Salicornia, which is the up intertidal species, and Spartina, you can, with previous studies on similar types of plants, we know that the roots are very stable. The biomass are very stable. They don't decompose that often. That's what makes Salicornia specifically would probably be a better phytostabilizer than Spartina due to the turnover rate just being much lower for the roots. And then the subtidal species, we looked at Zostra, which is a seagrass. Now, there were only previous studies on Zostra marina in Europe, and they didn't see much accumulation at all in Zostra marina. So they concluded that it's not really with it in terms of accumulation, but it could be a good bioindicator. Now, the southern African species, Zostra capensis, showed completely the opposite. It had good accumulation in its above ground organs and in the roots. However, the seagrasses have high turnover rates, so those sink of metals will only be seasonal, while the other 2 plants, the sinks will last much longer. And yeah, that's it from my side.
Thank you very much, Morelay. The final presentation that was done in the space of bioaccumulation is by Heinrich Dahms and co-workers looking at fantastic plastics
and their uptake by the African sharp-toothed catfish. Heinrich.
Good morning, everyone. So in our study, we tried to determine whether an African fish species was ingesting microplastics in our river system. We did know that from previous published material that we did have microplastics in the Vaal, but the problem with microplastic research just in general is we don't really have good biological indicators for their presence. And when we looked at the relationship between microplastics in the water column and in the sediment and in the fish, we could see that how it distributes in the ecosystem is quite unique in the various abiotic and anthropogenic factors of the environment can actually significantly change the amount of plastics that we can find there. Why this is a problem is because when we get to toxicological studies, the types of plastics that are used compared to what is found in the environment, you can't relate. You can't relate to how they've changed in the environment and just the environmental levels that specific animals might be exposed to. So that creates a bit of a problem in choosing a biological indicator firstly, and then where you would look into microplastic pollution as a whole, because how could you then describe a total amount of plastics that you might have in a specific section of a river system, or how much it might be expelling into the oceans? So that was the prime take-home message from that study and other components of the dissertation, and that's what I'm planning on looking into in the future. Thank you very much.
Thanks, Heinrich. Yeah, now once animals have been exposed to pollutants and they're taken up by the organism, those pollutants that reach the target cells interact with the target cells and elicit some sort of response. So the next 4 presentations were related to biological responses of aquatic organisms to exposure to different pollutants. The first presentation was by Tendo Muchakewa and colleagues, and it was looking at mortality and behavioral responses of Oreochromis mozambicus to 3 commonly used macadamia plantation pesticides. Tendo.
Hello.
Hello, yes, please go ahead.
Okay, our study, we looked at the response of fish Oreochromis mozambicus to 3 commonly used pesticides in the macadamia orchard, which are Karat, Zion, Mulan, and Parnax, and we also did control which were pesticides free. The reason for this study is with the increasing decline of the aquatic system and increasing plantation of this macadamia, we therefore decided to study the effects of the pesticides which are used in the macadamia farm on the fish population in the river bodies. To do so, we conducted an experiment to investigate lethal dose 50, which is known as LD50. We further assess the behavior response of the fish as soon as they were exposed to these pesticides at different concentration. We found that fish which were exposed to the control, they did not exhibit any abnormal behavior. Therefore, the results from control were taken as standards for the entire experiment. Fish which were exposed to this pesticide at different concentration were characterized by slow movement at first and then active swimming, which were therefore slow swimming, similar to those in the control. And early onset behavior were observed at higher concentration, and delayed behavior were observed at low concentration. That's all from my side.
Staying with Oreochromis mozambicus, Ntombi Kasiye Sibeko and co-workers looked at the effects of an antiretroviral antibiotic mixture on some hematological parameters in the Mozambican tilapia. Ntombi, are you available online?
Good morning, everyone. My name is Indomitable from the University of Johannesburg, and my study investigated the effects of the ARV drug nevirapine and the antibiotics sulfamethoxazole and trimethoprim on the blood parameters of Oreochromis mozambiquus. This was an exposure study that lasted 28 days, and the results of the study show that long-term exposure of Oreochromis mozambiquus to the mixture of sulfamethoxazole, trimethoprim, nevirapine induces acute stress and also causes anemia in fish. Thank you.
Thank you very much, Ntomi Bukasiye. Moving on and staying with fish, but looking at one of the standard test organisms, the zebrafish, Daniorerio, Donovan Grant and co-workers looked at the toxicity of vanadium to these zebrafish. And as Donovan can't be here, it is always, if you're a co-author on the paper, it doesn't come with a free lunch. It means you need to say something if the main author is not available. So Richard, you're on the spot.
Yeah, thanks, Victor. My students seem to make a habit of doing this to me at least once in a conference. So yeah, what we did was we were looking at the effects of vanadium pentoxide toxicity on Daniorerio by conducting first both chronic and acute toxicity testing to work out the LC50s for both chronic and acute. And then we went on to expose the fish to 10, 15, and 20% concentration of that LC50 value that we had determined. And we found that vanadium, the LC50 for vanadium is actually higher than environmental relevant concentrations from the Nile River and the Vaal River that we've tested at the moment. And from that, then, once we had done the chronic and the acute testing, we found that the vanadium had a significant effect on the GST activation and MDA content within the fish, and then had significant impacts during the acute testing on the energy consumption of the organism. And in the chronic testing, we found that it had significant impact on the MT concentrations, which then shows that with the bioaccumulation studies that the MT is actually helping the fish to cope with the amount of vanadium being accumulated by the organisms. During the chronic tests, the MT concentrations were increased, or MT content was increased. Joe, let's just go with that. And the bioaccumulation concentrations within the fish in the highest concentration during the chronic test was actually the lowest. So it looks like the MT was doing what it was supposed to be doing and helping the fish to reduce the amount of vanadium that is accumulated.
Thank you very much, Richard. And to end off these individual level effects, we have the presentation by SASAqS and colleagues looking at deformities in Oreochromis mozambiquus following exposure to a pharmaceutical. Sasha.
Thank you. Good morning, everyone. My name is SASAqS, and I am a postgraduate student at the Department of Zoology at the University of Johannesburg. As said previously by Marie Clementine, the current focus of our lab is the effects of drugs used in the treatment of HIV and AIDS. This study looked at effervescence as it has been found in surface waters throughout the country. And in previous studies, it was shown to cause histopathological changes in the tissues of adult Oreochromis mozambiquus, but to our knowledge, nothing had actually been done in the effects of effervescence in the development of juveniles. So we exposed these juveniles from the embryonic stage until 60 days of growth, at which they were fingerlings. So from the results, we concluded that although the drug does not have teratogenic effects, it does affect the formation of bones during early growth. Thank you.
Thank you very much, Sasha. I would just like to make use of this opportunity and thank Sasha Ntombi Kasiye and Marie Clementine, for presenting following difficult circumstances with the untimely passing of their supervisor, Professor Ina Wagenaar. So our condolences to you, and we wish you all of the best with your studies further. We end off the feedback on the ecotoxicology component by looking at ecosystem responses. So I'm sure that Lumari, Chris, and Josie were wondering how the heck they ended up in the ecotoxicology session. And perhaps just to say that, well, everything that we do in terms of environmental or responses of biota to environmental changes can essentially be grouped under ecotoxicology. So if we look at some of the ecological, structural and functional responses, the first presentation that falls under this session is by Lumari Janse van Rensburg, and it's the use of stable isotopes to look at nutrient flows down the Kabompo River in Zambia. Lumari.
Good morning, everyone. Yes, thank you. My name is Lumari. I'm a master's candidate from the South African Institute for Aquatic Biodiversity, and my study looked at the Kabompo River Basin in Zambia, which is located in the Northwest Province. So essentially, we used a systematic literature review to try and determine which gaps in literature needs attention, and a large gap was highlighted by the lack of ecological data for this region, specifically relating to food webs for fish in the Kabompo. And because the Kabompo is the biggest tributary of the Upper Zambezi Basin, part of a bigger project for this region for sustainable management, we are looking at the specific river to act as a proxy with some baseline food web data. So after identifying that this part of the Upper Zambezi can provide us with good ecological information, we then used sampled tissues from fish, invertebrates, and vegetation material and stable isotope analysis to create food webs. So in the end, we're trying to create these food webs to show the passage of energy and nutrients through the system, and we're trying to determine the landscape scale processing that's happening here. Because of the COVID situation this year, results are still being analyzed. That's why preliminary data is being presented. So in general, we found that most of the information gaps to the Upper Zambezi was limited, especially to the ecosystem process data, but that these food webs from the Kabompo may act as a proxy to assist future management. All our preliminary food web biplots so far with stable isotopes show to be typical of what we expect for tropical fish food webs. And we are using omnivores and herbivores, the Labio cylindricus versus a Cynodontis species, to show a trend in the longitudinal nutrient contributions to the Kabompo and to possibly suggest the landscape scale process.
Oops, it seems that we've lost Lumari there.
Sorry,
oh,
can
there you go. your
I'm
last
back.
sentence, Lumari, if that would be fine.
Oh, my last one, sorry.
Yeah, I think we missed that one.
Okay, I'll just end off with saying we're looking at specifically nutrient processing in the Kabompo as a proxy for the Upper Zambezi, and so far the hypothesis is that we suspect the flood pulse concept and the riverine productivity model to be the main contributors. Thank you.
Thank you very much. The next presentation was by Chris Curtis and colleagues, looking at recent environmental changes and the paleolimnology of Lake Matatiele in the Eastern Cape. Chris.
Thanks, Victor. Yeah, so there were really two stories I wanted to tell. Very briefly, Lake Matatiele seems to have been overlooked. I'm fairly convinced from looking at aerial photography and other records that there's the site of a natural spring-fed mountain lake, which has subsequently been dammed around 1970. The relevance of that for this talk is that it provides a rare opportunity to do a paleolimnological study. So we obtained sediment cores in 2018 from this site in the Eastern Cape. And we use them for multi-proxy analyses. So we looked at trace metals, we looked at things called spheroidal carbonaceous particles, which come from power station emissions. We looked at diatoms, and we looked at pesticides, among other things. And what we found was that the sediments of this lake, which were lead 210 dated, so we take a sediment core, slice it into small intervals, date each interval, and we had a record going back to the 1970s. We used XRF and mercury analyzers to look at concentrations in the sediment. We did see a strong increasing trend in sediment concentrations of mercury and SCPs, which suggest that these are coming from a power emission source, which I think links nicely to Hannes's talk. We also looked at other trace elements, and we saw steep increases in things like arsenic, chromium, manganese, and cobalt since around the year 2000. The sediments were used as part of a master study, so datum analysis was done, which would be a whole talk in itself, but this seemed to show an increase in mesotrophic datum species over the last 20 years or so in the core, which seemed to tie in nicely with increasing sediment phosphorus concentrations. And these are thought to be due to local agricultural activities just upstream, although the lake is in a nature reserve, and there is a farming and livestock area just upstream of there. And there was a construction of a small farm dam just upstream of Lake Mataputini in about 2014. And then finally, we analysed organochlorine pesticides. Mark Humphreys at Wits did those analyses and found some steadily increasing trends in the sediment from the base of the core right up to the present day in the surface, but especially surface sediment, large increases in concentrations of, for example, DDT and its derivatives. So these are possibly related to this increase in agricultural activity, possibly also related to an increased mobilisation of sediment going into the system when the farm dam was constructed around 2014. To try and link this through to ecotoxicology, well, we weren't really looking directly at ecotoxicological effects, but we did compare the sediment concentrations with TEC, threshold effect concentrations, and PEC, probable effect concentrations. Most of the trace elements don't reach probable effect concentrations, but arsenic and chromium did exceed threshold effect concentrations where you might expect to start to see ecotoxicological effects on, for example, aquatic microinvertebrates in the system. So I'm really just trying to sort of fly the flag here for paleolimnology and show that it provides a useful tool, I think, for looking at baselines and trends through time in some of our aquatic systems. Thank you.
Thank you very much, Chris. Yes, indeed. So the paleolimnology does link through to ecotoxicology, as you correctly said. Well, so to end off, we have, and Josie did admit that she's doing a blatant advertisement for a new project coming up. But yeah, Josie, so if you want to tell us about Dirty, and I trust that you had your tea in your SASAqS mug. So.
Good morning, Vic. Can you hear me?
Yes, indeed.
Okay. Hi, everybody. Yes, so I'm Josie Pegg. I'm from the Inland Fisheries and Freshwater Ecology team at SAIAB. And most of the time I do work on inland fisheries and ecology. But I'm also quite interested in carbon cycling. And earlier this year, with some European colleagues, I published some old work I did back in Europe where we looked at carbon fluxes from small streams. Carbon is, it's big news at the moment, the amount of carbon we're putting into the environment. It's not an acute toxicity, but it'll probably kill us all in the long run. So it's really important to understand how our natural systems, as they are naturally or as they're impacted anthropogenically, are altering climatic carbon. And historically, freshwater systems, because they comprise so much less of the globe's water than marine systems, weren't really considered as an important source or sink of carbon. But we now know that although they comprise much less of the total water volume, disproportionately they emit a lot of carbon to the environment. Here in South Africa, most of our fresh water is actually in dams, but there isn't very much published on how dams act in terms of carbon cycling, whether they are sources and whether they are sinks. And obviously dams are highly variable, from little small farm dams to, you know, our huge dams. So I'm hoping to sort of use my experience from Europe, where we have these huge sort of large-scale collaborative projects, to actually run a project here using a very, very simple methodology to monitor carbon cycling, specifically sedimentation and decomposition rates in dams. I am looking for partners to work on this with me. If you see my talk, you'll see all the methodology, but the sort of novel aspect of it is that instead of using anything like leaf litter to study decomposition, we'll actually be using tea bags. There's some great work done by the Dutch, which uses this novel method. All the equipment will be provided to partners. All we need is people that are willing to sort of sponsor a dam of their own, put equipment out in between February and March next year, and then retrieve it 90 days later, and be involved in this big project. Please get in touch if you're in any way interested. One thing I would like to say is I'm at the sort of beginning of the project, and actually, if anybody hopefully we'll have this network of people sort of working on dams during a set time frame. So if anybody's thinking, oh, well, you know, I would love to work on dams to collect this information or this information, everybody here does sort of slightly different things. So if you would like to sort of piggyback a little project on that isn't going to particularly add to the expense, or that you can cover that, I'd be really excited to talk to anybody either that wants to take part. Please watch my talk and see what it would involve, but also if you think the opportunity of people studying dams could be great to involve your work, please talk to me about that as well. Thank you.
Thank you very much, Josie. That I think brings together a lot of our different presentations that were submitted for this session. What I'd like to do now is that, without necessarily going through the same order of people again, provide the opportunity, if there are any questions to any one of the presenters at this stage, and I know I can't always see hands that are raised because of the 84 plus people on the meeting. I can only see a very few of them. So please feel free to unmute and then to just give your name and then ask the question.
Okay, I think there was one question from YouTube that I'll just raise for Ntombi Kayise Sibeku, I think, on someone said it was a very interesting talk, and they were wanting to know if there's any evidence of antibiotic resistance in humans as a result of eating fish that have accumulated these antiretroviral antibiotic drugs.
There are currently no studies on that. However, there are studies that look at water as a source of antibiotic resistance.
Yeah, any other further comments on that? Perhaps if I could put a question to Chris Curtis. Chris, you mentioned that the pesticides, and particularly the DDTs and their different congeners are increasing. would this be related to the use for malaria vector control, which is done in northern KZN, and then just grasshopper effect, long transport over long distances?
Thanks, Victor. Yeah, I'm not sure in Lake Matatiele. I know there's been work done in the Maputoland lakes by Mark Humphreys and co. Looked at local sources of contamination by DDT. It does seem a coincidence to me that there's been this agricultural activity at Lake Matatiele in the last 5 or 6 years. So it could be local contamination. I don't know what the farmer would be using it for, but this nature reserve is far outside the town, so I don't think that will be for a malaria vector control, unless that's practiced for livestock as well, which it may be, I guess. Do livestock get malaria? So there is evidence that DDT and other pesticides can be transported long range. So look, it could be either sources, but just because of the timing of it, I'm assuming that this is a recent local source of addition.
Yeah, it's quite interesting. Chris, do you know what the ratios of the PP OP DDTs are?
I do have those data, Victor. I haven't finished the analysis yet. I actually got these data fairly recently. So on my talk, there is a plot. I didn't do the calculations to look at the ratios. I know it tells you about the degradation.
That's right. Yeah.
And the age in the sediment. So that is something that we'll look at. I'm trying to work this up for a manuscript at the moment. So, but thanks for the heads up. Yeah, that's definitely worth looking at more closely.
Thanks a lot, Chris. I see Nico Rousseau's hands raised. Nico.
Thank you very much, Victor. I've got questions for 3 presenters, and let me ask them and then they can respond. I'd like to start with Curtis that talked about paleolimnology. Do you think your work might be able to be helpful in climate change research to give us a baseline of what the far past might have been in some of our water bodies, our natural lakes? Okay, so that was for Curtis, and then for Josephine Pegg, I've got a question about her experimental design. Many of our dams are used for water supply and irrigation during the dry months, with the result that water levels fluctuate quite widely, especially when you've got drawdown during the winter months. When your experiment starts, and I was just wondering if you've got any suggestions on how one gets around that issue. And then lastly, on the Zambezi presentation by Laura Lomery, I was just curious if they are already picking up some impacts of agriculture in their study and of mining impacts. I believe that's quite a big concern in the Upper Zambezi. Thank you very much.
Thank you, Nico. Yeah, so if we can go in that order, Chris, if you would like to respond first.
Yeah, thanks, Nico. There's loads of potential in using paleolimnology for paleoclimatology. That's a sort of whole other branch. My studies have tended to focus on the sort of Anthropocene industrial sort of period. So lead-210 dating goes back about 150 years. You can use carbon dating and other forms of dating to go back beyond that. But certainly datum analysis, like some of the data presented in my paper, has been widely used to look at ecological responses to climate change, as well as to sort of ecotoxicological studies linked to, for example, acid deposition or trace metals. So lots of potential there, yeah, absolutely. But I think one of the big challenges globally in the science has been separating out the signal of climate change from the signal of, for example, acid deposition and atmospheric emissions, because widespread use of fossil fuels that emits pollutants like sulfur and nitrogen or trace elements like mercury is sort of coincident with the period of climate change as well. So there's been an awful lot of work done using isotopes, using diatoms and other proxies to try and tease out where changes are due to, for example, nitrogen inputs from atmospheric deposition, or whether they're due to warming and other effects due to climate change. But a lot of potential there, absolutely.
Thanks, Chris. Josie?
Hi, Nico. Thank you for your question. Yes, so the methodology involves taking sort of decomposition measurements actually at 3 depths. One measurement buried into the sediment, one on the sort of the bed of the dam, and then one sort of a floating measurement in the water column to get the sort of pelagic, what's going on in the pelagic. So it does cover both sort of very shallow water to a pelagic depth. We only need 1 meter of water. So unless you live here in Makanda and the dams are completely empty, it should be a methodology that could cover the vast majority of dams. But that certainly is something that when we're planning on which sites we actually select, we will take into account. And certainly my hope is that people will be working on a dam, as I'm not working there, as I'm actually going to be working with somebody with their local dam. I'm going to be working on the sort of local knowledge and letting people make their best judgment about that. But thanks for your question.
Thanks, Josie. And Lumeri?
Hi. Yes, thank you, Nico, for your question. So far, I can tell you from what we've seen, this is still ongoing now, but in terms of pollution, this river is very, very clean. Normally, if you'd look at South African rivers, the nitrogen content normally tells us how polluted systems are, and the results from the Kabompo River so far show almost no signs of pollution for trophic positions for the fish. And some other things that we've also seen, I've had someone working with remote sensing data that's actually made a land use map for me of the region, just as an addition to see what's happening. You can see some places here and there where the Muyombo woodlands that cover most of the Upper Zambezi are starting to thin out because of human activity. But as far as the Upper Zambezi goes, it's still relatively pristine, and I'm hoping we'll probably see more as we go on with our analyses. So far, it just looks like a really clean river and really healthy fish. Thanks.
Thank you very much, Lumeri. There's a question to Tendo from Simone Dams. It's related to the selection of concentrations used for the exposures. Tendo, what was the rationale behind the ranges you used?
Hi. Highest concentration in this experiment were raw concentration of the spray itself, which are applied directly to the macadamia plantation. These concentrations were provided by the macadamia farmers, and the lowest concentration were chosen based on background concentration levels that the water bodies are likely to have, and these ones we found through reading literature of similar studies, but in the environment.
Excellent. Okay, so the selection from based on the actual use is that the concentration that's applied directly, Tendo?
Yeah.
Okay. Right. No. Thank you very much.
There's also a question, 2 questions that was raised for Hannes that was answered in the chat. So the one was first on about the concentrations in the sharks, and does it exceed human health risk or dangerous levels for humans? Hannes indicated that most of the sharks have concentrations exceeding the guidelines for human consumption, but as many of the sharks are protected in South Africa, the consumption is generally absent or at least limited. And then there's a second question for Hannes on what spatial data in terms of mercury exists in coastal organisms. Hannes.
Hi, Curtis. Thank you. There's quite a lot of data available for for mercury in coastal organisms worldwide, but there's quite a limited amount available for South African species, especially in elasmobranchs. So there's only 4 studies been conducted in South Africa, one focusing on the mako shark in 1980s, and then there's 2 focusing on smoothhound sharks in Langebaan. And the only other paper that's available was from KZN's Sharks Board, that they looked at sharks that suffocated in shark nets and drum lines, and then they tested mercury concentrations in them. So that was conducted between 2005 and 2010, and until now there hasn't been a recent study following up on that. So for most of the stuff, there's also nothing available on skates and rays. That this is the first reported mercury concentrations in skates and rays.
Excellent. Thanks, Hannes. Tendo, just a question to you from Jeffrey Lebepe, and it's just about the sample size that you reported on.
Hello?
Yes, it's just a question from Jeffrey Lebepe, and who asks, do you think the sample size of 2 individuals per jar is sufficient?
Yeah, we had 4 replicants for each concentration, so I think it's sufficient.
Yeah. I think maybe, Tendo, what you might just need to do is, if you look at standardized methodologies and just for normal toxicity testing, usually the replicates are not more or not less than 7 to 21. Now, I know 21 is a lot, but it's perhaps advisable, as I think Jeffrey correctly points out, that an N of 4 is not necessarily the best representation. So maybe just look at it. I know the difficulty very often comes in the analysis of the residues, and that becomes quite expensive if your N is too large.
Okay, noted. I've taken note.
Thank you. Josie, a question to you from Lucien, and it's related to estuaries, and yeah, I don't know whether that's that's a private message, but maybe linking the carbon research results from the estuaries to the dams as well.
Right, yeah, there's a lovely presentation in the estuary section by Kezia. So I think it's more that there wasn't the possibility to work on everything. So I've just delineated myself to work on the freshwaters. But I know that there are people working on dams. Yeah, Lucienne, it would be good to talk to you. So yeah, it was really a case of limits of my capacity more than anything else. But yeah, it would be good to talk. So thank you.
Right. I've got just a question to Heinrich related to the microplastics in the catfish. Heinrich, did you guys run a environmental blank together with the sampling and the extraction? And to what, if you did, to what degree is there external contamination? The reason why I ask is that we've actually found that there is quite a lot of external contamination when doing the microplastics dissections in the field and then bringing them back to the labs.
Yes, Prof. We did indeed. Contamination, like you said, it's a big or massive challenge with microplastic research. So we tried to do our best to prevent this. We took environmental blanks. from collection all the way through the preparation of the samples, during digestion, during reading. And then at the end, we did count the control samples to see then if we had contamination. In the water, we didn't have too much contamination because 90% of the time, the jars or the containers that the water's in is kept closed during reading, during processing. For sediment, it was a bit more trickier because obviously when you dry the sediment in the oven, you can't completely cover it because you need to get rid of the moisture. So we did pick up in our sediment blanks, we did find some fibers from the air, probably from the dust. And then, yeah, obviously we took, or I took just a roll call of clothing that's worn at the time, the color of the lab coat, the color of the gloves, and then at the end, anything that resembled something that we wore or some of the material that we were used was then removed from the data set so that we can get just a better representation of what it would be like without some of the contamination that we did find.
Excellent, thanks, Heinrich. Yeah, as you rightly comment, it really is a sort of a conundrum when you're looking at microplastics, and very often at a lot of the other micropollutants as well, is that the air around us has so much in it that we might be over analyzing or overestimating the level of contamination, and therefore these quality control measures are extremely important. I see we've reached 12 o'clock. Are there any other questions, burning questions that people would like to raise to the presenters or any one of the presenters? I know that each of these presentations do allow for many questions. If not, then I'd like to thank all the presenters in this session and invite the participants, conference participants, to, if they have not watched the presentations, to please go and have a look at the presentations and vote for your favorite presentation. Get the family to vote for the favorite presentation. Wynand, yeah, from my side, thank you very much. I think this has been an interesting session with interesting questions. Thank you.
Yes, thank you, Victor. I agree. A lot of interesting talks and really good research and results that we've seen in this session. So I'd just like to echo Victor and encourage people to go watch and support all these 12 talks. There are really some good talks there, so please go watch it, like it. Don't dislike it, please. Just like it. And then that will be the end of the session. So we will break now again for lunch. So normally lunch is pretty quick for people if you're at home. So once you're back, please go watch the videos for the next session. That's starting at 1. Get your questions ready, submit them on YouTube. The second session today will focus on aquatic invertebrates. And again, please log in 10 minutes before so we can start on time and have some active discussions in the next session. So if there's no other burning questions for the speakers or on housekeeping, we'll see you back here at 1 o'clock or just before 1. Remember to register for the SASAqS medal ceremony in the link that was sent as well, please. And thank you for this first session. I think it really went well.